Good evening. Welcome to People's University Livestream Science. This is part 2 of chemistry. Before we get to our guest tonight, um, yeah, I'm getting a little feedback there. Okay, thank you. Let me just make a couple of announcements. First of all, next week on People's University Livestream, Dr. Stacy Cafalos Vargas will join us. She is another Wheeling College graduate and a friend, and she happens to be a physics professor at Virginia Military Institute. Her class will be the fundamentals of physics from Newton to Einstein, so don't miss that. That's next Thursday at 6:30. This evening, I'm sorry, before I get to that, next Tuesday, February 9 at noon, our Lunch with Books live stream will feature Jill Newmark, a historian talking about African American Civil War surgeons. Now, as I started to mention, we're talking about chemistry again tonight. I wanted to let everyone know that there was a program on Nova PBS last night called Beyond the Elements Reactions, which kind of is a good supplement, I think, to what Erin was teaching last week. So you can go to PBS online and watch the video for free, or if you have PBS, you probably get it on demand. This evening is Chemistry 2. Now, here's my introduction. I've been organizing programs for about 15 years now, and I've always wanted to have my sister on. I never thought I'd get the opportunity, but I just wanted to say that I'm very proud of her. I've watched her evolve from one of the smartest, hardest working students I've ever known. I mentioned Wheeling College, but it goes back to Wheeling Central, Corpus Christi, at every educational level, into an accomplished elite scientist on the front lines of the fight against one of the most daunting existential threats to humanity, and I'm not exaggerating. We hear about the virus pandemic, but antibiotic-resistant bacteria is just as dangerous and just as much of a threat to humankind. We have to remember bugs like MRSA, and she'll tell you all about that, CRE, ESBL-E, and the BL. The list goes on, the alphabet soup of bacteria. I am proud to say that my sister has done cutting-edge research on that front for many years now, and we're lucky to have her powerful brain working for us, and she and her colleagues give me hope. If our species is to survive its own folly, science will lead the way. And that's part of the inspiration. I believe that for this series. My sister is a scientist and a very good one. I used to call her an egghead and tease her about her eschewing the humanities, but that was in good-natured fun. She's earned my sincerest respect, and I'm honored to have her here tonight as instructor for this series. Here is Dr. Erin Duffy.
Thank you, Sean. That was entirely too kind, but I'm honored to be here. I have to say this is a real treat for me, and I'm really looking forward to the next hour, and definitely also looking forward to the next 2 weeks with Stacy. Tonight's presentation is going to be a little different than last week's. It's going to be a lot more applied because I really did want to introduce you to the topic of drug discovery briefly, how we apply it to antibiotic drug discovery, and then talk about why we need antibiotics. And so I hope you'll find this interesting. And, you know, if you want to talk more in depth about how to do drug discovery and the chemistry role in that, you can always come back for another. Okay, so I am going to share my screen. I hope this is going to work. Yep. Okay, so hopefully everyone can see this. Okay, so, you know, I always have to start when I'm talking about my work in industry with this sort of disclaimer. So for those of you who aren't familiar with the process of drug discovery and development, I thought I'd give you a sense of what's involved and the timeline. And this is true for drug discovery, irrespective of therapeutic areas. So whether it's designing and developing a new cardiovascular drug, a new antibiotic, or as we've all seen play out here in the last year, a new antiviral agent. So it all starts on the left with the identity of the disease you want to treat. And then there's a lot of effort to try to determine whether there's something, a particular target in the body.
Sorry to interrupt. We're not seeing a slideshow. We're just seeing the.
You're not?
No. Try running the slideshow from the beginning.
Okay. Does that work?
No. We're still seeing the preview.
Oh. Okay, should I'll unshare and share again?
Try it again, yeah.
Okay. I don't know how to do that. All right, let's see what happens here. Share. Now what happens?
Well, now we have the preview again. We can see that on the screen, so, but the slideshow itself. There. Well, almost. Now it's just full screen.
How about that?
It's still just the preview, but we can see the slide if you want to just click through the slides that way.
Okay.
I'll go away.
Is it okay? So you can see this okay?
We see your first slide, but if you're going to...
You don't? Oh, that's terrible. Okay, I really don't know what to do about that. I don't know why it worked last week.
Yeah, it did work. I don't know what the problem is either.
Okay. Can you see this okay if I click through it like this?
Yeah, I think we see pretty much the full slide there, but you know, we see the sort of toolbar at the top, but that's okay.
Yeah.
We can also see the slide, so you can
Okay.
do it that way. I'm sorry.
Yeah, no, sorry about that. I don't know what the deal is. Okay. Well, let's give this a whirl. So anyways, I was saying, you know, we start always with identifying the disease you want to treat, and then trying to identify and isolate a target within that. So an example would be, you know, you've heard a lot about people and the pursuit of an Alzheimer's drug. There have been a lot of failed attempts to find the right target for that. You may have heard about amyloid and other targets like that. And so people try to identify things that they believe through some, you know, biochemistry and also molecular biology, when you turn them on or turn them off, have an impact on disease. And so once you identify that target, then you spend a lot of time doing chemistry, biochemistry, molecular biology, and design to try to make molecules, like we talked about last week, that will bind to and either inhibit the function of a target that will have impact on disease or enhance the function of a target. And so it depends on what the goals there are. Once you've done that, and that's a lot of laboratory work, then you'll want to demonstrate that it actually works in an animal. Now, here's a big thing. A lot of therapeutic areas, the animal models are not good. And so you have no guide from the laboratory test tube to really the human in terms of whether what you're designing is going to result in some impact on disease. Fortunately, in the area of infectious disease, and especially true in antibiotics, mouse models of infection are absolutely predictive of what you will see in man. You might have to learn what levels you need to achieve in a mouse in order to correlate with the clinical outcome, but you have a much better sense that what you've done in the laboratory is going to translate into humans. You then have to make a lot of the drug that you've decided is the best, and you'll have to formulate it in a way that can be delivered most expeditiously and appropriately for the infection. So if you have an infection in the hospital, and let's say that you're, you have an IV line anyway, maybe you're critically ill, an intravenous formulation of an antibiotic is perfectly fine. If you have, let's say, a pneumonia that you acquired from the community, you might start in the hospital, you might come to the emergency room, but you're going to want to get out of the hospital, and the hospital is going to want to get you out of the hospital. And so to have an oral formulation, either in a tablet or a capsule or something like that, so that you can take it home without having to have a PICC line installed, is really critical. And this can actually take a lot of time. And this comes back to, we talked about last week, the molecular properties of molecules. So things like solubility, things like permeability, stability of molecules, these things really matter in terms of the flexibility you have to formulate them for use in humans. Then there's a whole series of human clinical trials here, and typically there are 3 phases of human clinical trials. The very first is what they call phase 1, and this is a study usually in a small number of healthy volunteers, so 25 to 30 maybe, typically aged 18 to 55. And in that study, what you're looking for is, do I achieve in the plasma, in blood, the drug levels that I think are appropriate and model back to what I see in the mouse for efficacy, or whatever species the efficacy studies are done in. And at that level, is it safe and well tolerated? And so that's all you're looking for there. Once that study is done, and assuming that you have a good outcome there, then you go into what are called phase 2 studies. And this is the first demonstration of efficacy. So now you're looking in patients in whatever indication you're going for. So for instance, if you have a drug that treats bacteria associated with hospital-associated or ventilator-associated pneumonia, then those are the patients in which you're going to test this drug. And typically, your phase 2 trials are done in maybe 100, 200 patients. Often there's more than one arm. You might be testing more than one dose level or more than one dosing regimen. Is it one day, once a day, twice a day, etc.? And most often you have a comparator arm of an antibiotic that is known to work, so that you can have a benchmark for at least non-inferiority, and then assessment of whether there are features that make your drug even more attractive. The final stage of clinical trials is called phase 3, and that's just phase 2 on steroids. So typically this will be several hundred patients, generally worldwide, so that you have the best understanding of how well your drug performs in multiple situations. Assuming all that works out well, you file a document with the regulatory authorities here in this country. It's the Food and Drug Administration. And then they evaluate the entire data package, everything that you've done in the preclinical space and everything in the clinical space. And then they make an assessment on whether they feel this should be approved for use in humans. Once that happens, then, or actually in parallel usually, but you are bulk manufacturing so that you can distribute your drug, in whatever jurisdictions it's available, so that it can do the best good for patients. And of course, in every area but antibiotics, you care about sales and marketing. Okay, oh boy. So this is a slide from last week, but I'm bringing it up again. We talked at the end about something called non-covalent interactions, and I used this terrible graphic the last time, so why not do it again? These are not like the covalent interactions that make up molecules, but rather these are interactions between molecules, not reactions, but interactions that are often called binding. And so these are typically in equilibria, meaning you'll have, in this case, let's talk about a protein target that is relevant for disease and a small molecule that you've designed to bind there. And there's some attraction that makes that favorable, but they're not reacting, so they can't come apart. The strength and relative strength of these interactions can be evaluated in the laboratory so that you understand what that interaction is and how you might enhance it in new molecules that you could design. So, okay, so it binds the target. Am I done? Well, the answer to that is no. And so we talked a little bit about this in the graphic a few slides ago. But coming back to this idea of intrinsic molecular properties, we talked about things last week like boiling point, melting points, solubility, and there are many others that govern whether something that inhibits a target actually becomes a drug. So the molecule that you've designed has to actually get to the target. Often this is intracellular, so you've got to demonstrate that you have the properties to get into cells. The molecule then has to stay in those cells. So cells, bacterial cells and human cells have basically a bunch of pumps that recognize a wide variety of properties, and they're actively trying to get you out of the cell all the time. And so how do you build in the properties that avoid getting ejected from the cell? That's called efflux. And you also have to show that it works in an animal like we discussed, and not just in a test tube. And features that govern that, again, are stability, solubility, and other things that we'll talk about. And then finally, that molecule that you've designed should be selective for the target that you want and safe, which means often doesn't bind to other targets, doesn't cause toxicity, and can be used safely. So the question is designing an antibiotic the same as other drugs? In many ways, yes. Again, all those intrinsic molecular properties remain important. But in addition to that, if you're thinking about, again, a cardiovascular target, you have to get into the body, and not be ejected out. But then you go straight to the human target. With bacteria, you got to get into the body. You got to go to the organ where the infection is. Sometimes it's in blood if it's sepsis. Other times it's in the lung. It's in the urinary tract. It's in the skin, etc. And so you've got to get into that environment successfully, and then you have to get into the bacteria successfully. And the bacteria, depending on which bacterial species it is, the bacteria can have multiple of these so-called efflux pumps. One of the toughest bacteria to treat in pneumonia right now is something called Pseudomonas aeruginosa. A lot of cystic fibrosis patients suffer from that. And Pseudomonas has something like 30 plus different efflux pumps just waiting for your new antibiotic to try to get into the bacterial cell to get to its target. And before it does it, it kicks them out. So you have to understand how to avoid all that and balance that with all these other properties. And then finally, if you're successful there, you got to find your way to the bacterial target and bind as you're predicted to do. So in terms of what happens in the laboratory then these days, everything starts with molecular design. And this is largely a chemistry exercise. So we talked about last week, and I attempted to share with you a movie which was the atomic details of the ribosome. By the way, the ribosome is a very large antibacterial target. And so what you'll do is you look at these structures at the atomic level, you know where you need to bind to inhibit its function. And so then you use computational algorithms. Someone asked a question last week about using artificial intelligence, in some of these exploits. And this is exactly what you do. And you try to predict how these molecules will interact. And also you'll try to predict the properties of these molecules. And once you then design those, often what you'll do is you'll choose initially a set of maybe 15 to 20 different molecules to make. If you're wise, what you'll do is you'll select some that you think should bind really well and others that you think won't, for the purposes of learning your bounds, what's good and what's bad. So then chemists who do molecular synthesis will then make these molecules, and there's a lot of creativity involved there in terms of defining reactions that will deliver molecules that no one's ever seen before. And then once you make them, then you test. And initially, you're going to test for this on-target activity. Do you bind to the thing that you want to bind to? And is that a favorable process? Do you get into bacterial cells and cause them to die or weaken? And are you not broadly cytotoxic? So are you specific for the bacteria? Often this takes several iterations. In fact, it can take years. But when you've satisfied these initial criteria, then the next step you want to ask are things, are you efficacious in a mouse? But before you do that, you just want to give the mouse a little bit of drug, maybe march it up to where you see tolerability signs so that you have a sense of where to start looking for efficacy. And then you look at these properties to ask, is it going to be compatible with getting into the body, whether it's an animal or a human, to cause this effect? So these things are all done. And often there's an iterative process here as well, because now you're making trade-offs, often activity versus efficacy, and where's the balance there that you can live with. And once that's all set, then you advance to a whole series of studies that looks at advanced efficacy, maybe in more than one model, looks at things like resistance development. We're going to talk about that in a few slides. And then importantly, toxicity, so that you can prepare the best package you can to gain regulatory approval to move into human clinical trials. So I mentioned that there are different types of bacteria, and broadly there are two categories. There's so-called gram-positive bacteria and gram-negative bacteria. And the reason they're called this isn't because it has anything to do with weight, but rather there's something called the Gram stain. And for those of you who have had a medical background of some sort, hopefully you'll recognize this. But so you can tell right away when you take a sample from a person, and if there's a bacterial isolate in that sample, if this is someone that you think has an infection, there's a staining procedure where if it's a gram-positive bacterium, it'll turn purple, and if it's a gram-negative bacteria, it'll turn red. And so you'll be able to see, hopefully at least what general category these bacteria happen to lie in. Then they all have interesting shapes that you can see in a microscope, and so you can actually tell a lot by their shapes as well. But the main difference between gram-positive bacteria and gram-negative bacteria is that gram-negative bacteria actually have two membranes before you get all the way into your target. So your target's like down here somewhere. So you've got to cross this so-called outer membrane, and this is tough. Okay, this is really tough, really tight junctions. And there's all these efflux pumps sitting just below it if you happen to get in there. And then you're going to go through a second bacterial membrane, and then once you do that, you get to your target. Over in your gram-positives, you don't have that second membrane. Now there's a middle layer, the membrane, the outer membrane of the plasma membrane that's thicker, but it's not the same as a double membrane. So it's generally easier to get into. And so again, part of the design here is understanding what properties allow you to get into both membranes, or if you want to target gram-positive bacteria in specific, then maybe you don't need to worry about that so much. Okay, so then let's see here. Okay, but unlike let's say COVID or even gonorrhea, which is bacterial infections, many of the infections that people get, whether it's pneumonia or a urinary tract infection or an acute bacterial skin and skin structure infection or a prosthetic joint infection, these can be caused by more than one kind of bacteria. So I'm just showing you here a little pie chart. This is a study that was published in 2016 that shows you the variety of bacterial species that may cause, in this case, hospital-acquired pneumonia. And so you see here, the biggest causative agent is Staphylococcus aureus. And of course, MRSA is a drug-resistant form of Staphylococcus aureus. The second largest is Pseudomonas aeruginosa. So about 30% of the hospital-acquired infections are staph. Now, almost 30%, 25, are Pseudomonas. Now, in terms of gram-positive, gram-negative, Staph aureus is gram-positive, Pseudomonas is gram-negative. Then you have a bunch of other players here. Klebsiella pneumonia comes in at about 11%. That's a gram-negative. E. coli and other like bacteria in that species come in at about 8%, also gram-negative. Acinetobacter infections, you do see these used to be something people didn't worry about because Acinetobacter is actually a pathogen in the soil that actually is good for soil. But it turns out it became a problem largely in the Iraq War, of all things, when your soldiers running, on the ground. There were bombs that blew. There were terrible skin infections, deep wounds. They were put on hospital ships where they had inadequate supplies of good antibiotics, and these infections festered. So you had all the soil, sand, whatever, that blew up in these infections. And that then caused the rise of drug-resistant Acinetobacter infections as those soldiers came back home and went to long-term care facilities in places, all over the country. Then they started spreading Acinetobacter, and this is now a big worry in our hospitals today. So that's kind of small. I'm going to make this figure a little bigger, hopefully, so you can see it. You don't need to see my title. So in terms of targets, there are many, many different ways that a molecule can inhibit bacteria and therefore be effective against infections. And so there are some that target the cell wall. So for instance, penicillin, cephalosporins, these are all in the same class. They inhibit a certain process in terms of making cell wall. Vancomycin, I'm sure everybody's heard of, does it differently, but also targets cell wall biosynthesis. There's also cell wall integrity as a target, and that's more about the structure of the cell wall. Then inside, you have some big targets. You have DNA synthesis and DNA gyrase, more broadly, DNA biosynthesis. And this is where your fluoroquinolones like Cipro, that's how they act to kill bacteria. There's also RNA synthesis, and there are classes of drugs not used much these days because of resistance, but rifamycins. Here's that ribosome that we talked about last week, whose role is to make more protein and therefore make more bacteria. And there are many, many classes of antibiotics that exhibit, that inhibit the function of the ribosome by binding there. And so these are, antibiotics that I'm sure you know well. So erythromycin, azithromycin, trade name Zithromax. Chloramphenicol is one that's not used much now because of safety. Clindamycin, This is used, I think this is sort of like #1, use it when someone has an infection, and remains kind of robust, but there's resistance to that too. Then there's the class of tetracyclines. There are also aminoglycosides not mentioned here. And then finally, just inside the membrane, there are classes of drugs that just serve to sort of disrupt it. And these are classes of molecules called polymyxin. In fact, in that class is a drug called colistin, which is frankly rat poison, but it remains the last line of defense for some infections, and so it is still used today. So I just thought, I'd show you, I'm going to have a little focus here and there on penicillin, because of course penicillin is probably one of the first antibiotics, actually not the first ever discovered or used clinically, but certainly one that put the era of antibiotics on the map. And so penicillins act by binding to something called penicillin binding proteins. It was named much later when they mapped where penicillin bound and tried to figure out, what it was influencing or inhibiting in terms of stopping bacteria from working well. And so penicillin binding proteins, as I said before, are involved in bacterial cell wall synthesis. In fact, they're in the final stages of sort of stitching it together. And when penicillin binds, and it binds in this domain, there are 3 domains of a penicillin binding protein, it actually shuts that function down. Okay, but just as there are many ways that you can inhibit bacteria, there are many ways that bacteria can fight back. And so there are 4 key ways shown here. So some bacteria and things like Pseudomonas I've mentioned and Acinetobacter are among them. They just have natural resistance because their outer membrane is constructed so tightly that it's very challenging for antibiotics to actually penetrate that and get in to bind to the target and inhibit bacterial growth. There's also a lot of drugs that are impacted when the target itself is modified. And so I like to describe this like, you have a binding pocket and then someone sticks an elbow in it. So the synthesis, when the ribosome makes new proteins, the bacterial ribosome says, you know what, I'm going to put something a little different in here, and I'm going to change the shape of that binding pocket so that the drug no longer binds effectively. And this is what renders classes like the macrolides, so erythromycin and azithromycin, the quinolones, etc., ineffective. I've mentioned the efflux pumps that pump antibiotics out. They are broad, they are numerous, and they are pretty indiscriminate. And so they recognize a wide variety of chemical properties and kick them out. And then finally, there's a mechanism of resistance where the bacteria produces an enzyme that inactivates or modifies the drug itself. And that is what happens with the penicillins and cephalosporins. So what happens to penicillin in this? So there's this enzyme, it's a type of protein called a beta-lactamase. So penicillins broadly belong to a class of drugs called beta-lactam antibiotics. And so the beta-lactamase means it's going to come and chew it up. And so what it does is it focuses on this ring. This is a beta-lactam ring. This is the whole molecule of penicillin. And basically what it does is it just clips this bond, and then it opens the molecule up, and the molecule becomes a lot floppier, and it just doesn't fit into the binding site very well anymore. And so it's not effective at inhibiting the penicillin binding protein. Well, it turns out from a chemical point of view that there are many beta-lactamases, and they can do this reaction several ways. So there are whole classes of beta-lactamases that are, they use a so-called serine, so an amino acid that has an OH group here, and that combined with the water does the deed of attacking and opening this ring. You can also have types of beta-lactamases that are called metallo-beta-lactamases, because rather than using, this serine type group, it engages a metal group to then do the attack. So you can achieve this a few ways, which is a little bit frightening. And it turns out it's not like there are just a few beta-lactamases. So it actually started much earlier. So, penicillin was discovered in the 30s, and we'll see that on the next slide, I think. But you start to see the number of beta-lactamases really take off here, certainly a step change in around 1990 to almost 3,000 different beta-lactamases today. And there are several classes of them, and they act in different ways. And this is just showing you the distribution of the classes. But the integral over all of them means bad news. Well, maybe it's just a penicillin thing. We don't need to worry about it. There are lots of other antibiotics. Well, it turns out it's not true. And this is where it's a shame that I can't build this. But let me try to walk you through this. Okay, so here is time. Okay, and above the timeline is when a class of antibiotics was introduced for clinical use. And below the timeline is when the first instances of resistance were seen. Okay, so let's start with sulfonamides. Sulfonamides were introduced into clinical practice in, I think, 1936. And just about 1940, so 4 or 5 years later, is when the first clinical cases of resistance developed. Penicillin was first used clinically in 1943. It was actually found in Dr. Flory's coat, if you've read the book, around 1930. People were, goofing off with it in the lab and using it for emergency use, but it wasn't approved for clinical use until 1943, but there was already resistance to it in 1940. Here comes the aminoglycoside. So if anyone has taken doxycycline is a tetracycline. I'm going to come up with aminoglycoside here in a minute, but I've just lost my mind there. But anyway, aminoglycosides here, streptomycin is an example, were identified for clinical use in 1945, and that same year, resistance was seen to them. Here comes chloramphenicol in around 1948 and 1950, resistance. Your macrolides like erythromycin, also introduced in 1950, and they hung around for about 5 years until resistance developed there, and so on and so on. So you see tetracyclines, resistance was there before they were introduced. Here comes vancomycin in around 1958, resistance seen by 60. MRSA, which is officially called methicillin-resistant Staphylococcus aureus. And so methicillin, of course, is a type of penicillin. That first showed up in 1960. MRSA today really should mean multi-drug resistant Staphylococcus aureus, because it is often resistant to more than one class of antibiotics. Here come the fluoroquinolones. So this was a big deal, actually. There had been this major flurry of activity, as you can see, with the introduction of many, many classes of antibiotics, very successful. And then things started to drop off, I think, because people were thinking maybe, we didn't need so many more antibiotics now. We had a good arsenal. And the fluoroquinolones were actually first introduced from a laboratory accident. Someone was trying to make quinolines, which have nothing to do with antibiotics, and the reaction didn't go according to plan, and so the quinolones were made, and then the fluoroquinolones came after, and literally just stuck a fluorine onto the quinolone, and they're were born the fluoroquinolones. Very useful class of antibiotics, but also resistance appeared at the same time as it was in clinical use. And then there was nothing. For about 15 years, there were no new antibiotics that were introduced. And so while that was happening, the bacteria were just, sort of getting smarter and smarter and smarter. And so then people started to do variations on the penicillin class to try to make them more robust, to the inactivation by the enzyme. And there were two classes introduced, the carbapenems, and I'll show them on the next slide and how they're different, and the cephalosporins. But it didn't take too long for the bacteria to figure out how to inactivate them as well. And then there was another gap here in time of about another 15 years where there really weren't new antibiotics introduced until this new class of so-called oxazolidinones was introduced. And many of you probably know this is a very valuable drug, linezolid. But within 6 months of the introduction of linezolid, resistance arose. And then finally, you may know this as cubicin, also known generically as daptomycin, as a new class of lipopeptides, a variation on a theme of vancomycin, introduced in 2003, and resistance appeared in 2005. So again, very strong response from biology to chemistry, rendering all of these classes of limited utility across the board. So what does this mean for human health? Sean said this at the beginning, but this is a big deal. So, bacteria, I guess the point is resistance is a fact of life. And it's not that all of a sudden resistance is here. Hopefully you've seen that it's just been a drumbeat, which is why innovation has to continue to happen. Turns out annually, today, over 700,000 people die of antibiotic-resistant bacterial infections. And the sad truth is that there's only been one new class of antibiotics introduced and approved for gram-negative bacteria, which are what are causing a lot of our very serious hospital-acquired infections since the introduction of the fluoroquinolones in 1962. And so just to give you a sense of the rates of resistance for different classes, you can see that over time, so time is on the x-axis here, and percent of samples that are resistant are on the y. And you can see, for instance, the quinolones here, fluoroquinolones like ciprofloxacin and levofloxacin, there can be as much as about 22, 23% of all samples that are resistant to these drugs. And you can certainly see, many classes, again, with resistance rates around 10%. That's pretty scary, actually, if you think about it. So you have a 1 in 4 chance of having an infection that isn't going to be treated by the quinolone antibiotics. I can tell you in the area of gonorrhea, which, people are probably wondering, like, gosh, we haven't heard about gonorrhea in a long time. Well, it is back. It is back in a big way. And today there is a single antibiotic, not a class, where you have several examples that maybe are a little different and maybe will, represent a little opportunity for cure. There's only one antibiotic that is robust in the face of drug-resistant gonorrhea, and it's a very old antibiotic in the cephalosporin class. And when and if that goes, we're in trouble. And so a lot of people are now turning their attention both in vaccines to prevent gonorrhea, but also with new agents in new classes to sort of scramble the bacteria and make them think about how what they would have to do to become resistant to them. I'm not going to read this to you, but everybody is recognizing that this is a real and present danger and will be into the future. And so the CDC here in the US in 2013, and then again last year or 2 years ago now, issued their so-called threat lists. These are lists of bacteria that they're very concerned about because we either have very few antibiotics left to treat them, and in some cases, even none to treat them. And so Sean mentioned some of these earlier. So carbapenem-resistant Acinetobacter and carbapenem-resistant Enterobacteriaceae. So these, again, are these penicillinase-type enzymes that target carbapenems rather than penicillins, but structurally very related. And, you know, when you see these, big trouble because there's not much to treat them. Everybody, I think, knows about C. diff and recurrent C. diff. And again, very few treatment options there. And as I mentioned, drug-resistant gonorrhea is a big problem. And then there are the serious threats and the concerning threats. And then, you know, just our luck, there are some things that are coming on the screen now that people haven't really thought about or worried about. Certainly, when's the last time you worried about pertussis? Probably not for a long time. And Mycoplasma genitalium, which happens to run around with Neisseria gonorrhea. So it's the same patient population with very serious drug resistance rates. And the WHO has listed their priorities in very similar sets. In fact, they're almost superimposable of bacteria that cause serious infections for which we have very few antibiotics. So what can be done? Well, one thing, coming back to the penicillins and those classes more generally, is that there have arisen molecules called Betalactamase inhibitors. So these are little molecules that sit where the enzyme works. And again, that enzyme, you know, is meant to call the penicillin or, you know, cephalosporin or carbapenem in. They then do the reaction, breaks open the ring, and then it can't bind to the penicillin binding protein. Well, people have figured out at the structural level how to design molecules that don't have antibacterial activity themselves. Their sole role is to bind to this enzyme that renders the penicillin-type molecules ineffective. Now, this has been around for a while. So clavulanic acid was the first beta-lactamase inhibitor ever created. I'm sure many of you have heard of augmentin. Clavulanic acid was paired up with amoxicillin to restore the utility of amoxicillin by binding to and inhibiting these beta-lactamases, allowing amoxicillin to go about its business and do its job and kill bacteria. There are a lot more efforts in this arena. There have been 2 new programs introduced clinically recently that employ this strategy. There are also novel penicillin binding protein inhibitors that don't look anything like the penicillins that people are working on and are advancing through preclinical and hopefully clinical trials soon. And then there are people working on all those other mechanisms that we talked about and also making new classes there. Again, the whole purpose is to have a lot of different ways to confuse the bacteria. So in the 10 minutes or so that I have left, I'm sure you're all sitting there saying, oh, you know, I mean, we've been through all this craziness with COVID. This sounds really scary too. Please tell me there's a lot of active research in this area. This is kind of a bad news, good news story. So let me start with the bad news first. So the bad news is this is a plot you can't read, but on the x-axis is time starting from February of 2015 to July of 2019. And on the y-axis is U.S. sales in millions of dollars. Okay, and so you can see these are just all different bacteria. They almost all basically make no money. And there's, you know, one up here, and this is Vabomere that happened, or this Tuflaro actually, which is a new cephalosporin that makes about, it's been pretty flat, but about $12 million annually. Why am I telling you this? So you watch TV, you see these commercials for a drug called Humira. Everyone has seen these. Do you know how much money Humira makes a year? $18 billion, not million, billion. And by the way, the cost to get one of these molecules onto the market is somewhere in the area of about $800, $900 million over about 10 to 15 years. So that's a massive investment that you will never recoup at $12 million a year. So the net result of that is that all the big companies have really exited the space. They are no longer deploying researchers like myself to do antibiotic research because it's not profitable for them, and they can work on cancer drugs where you can charge whatever you want. and usually you don't even have a cure, as well as your more lifestyle-oriented drugs. And the reason for that, cardiovascular meds, depression meds, etc. These are things you're going to take chronically. You don't have to charge a lot of money for them, but people are taking them for the rest of their lives. So 2 ways to win there, but people are never going to pay that kind of price for antibiotics. And then the third part of that is that there are a lot of little companies that stepped in as these big companies were coming out. I worked at this one. I spent 17 years at this one. And in 2017, we received the approval for our first antibiotic that we took all the way through the clinic. And 1 year later, I laid off my entire R&D staff, and a year after that, the company filed for bankruptcy. That was true for this company. 6 months after they put their first antibiotic on the market, they filed for Chapter 11. And this company also was bought basically for no money, and all of its researchers laid off after they introduced their first antibiotic. Okay, but that's the bad news. The good news is that there are organizations like the one for whom I now work that are filling this gap. So what is CARB-X? CARB-X stands for Combating Antibiotic-Resistant Bacteria, and the X is meant to stand for acceleration. We are a global not-for-profit partnership. We sit within the Law School of Boston University, and we were created in 2016 to do just this, support the early discovery and development of new products to diagnose, prevent, and treat bacterial infections. And so we do that. Again, so this is our mission to stay prepared and have an arsenal of antibiotics in the face of drug resistance. And we do this by supporting little and big companies. We'll support academics anywhere in the world. If you have an innovative idea, we support it. And we take that idea all the way through that first phase of human clinical trials so that they can demonstrate safety in a human. And the idea is that hopefully there will be groups that take it on for advanced development. We are funded by 3 international governments and 2 foundations. So you've heard probably recently a lot about BARDA and ASPR. These are the people behind, the COVID vaccine rollout, Operation Warp Speed. The UK government and the German government, these are all supporters of ours, and then the Wellcome Trust and the Bill and Melinda Gates Foundation. And of course, the NIH, and the NIH is very famous now because of Dr. Fauci. He's a household name. They support us through a series of very important so-called in-kind services. So they will do toxicology studies and efficacy studies for companies of ours working on new antibiotics, and they do this for free. So they don't give us money, but they supply these services, which is terrific. Give you a sense of the magnitude, we have $480 million to support antibiotic research. I'm not going to go into all of it, but I just wanted to give you a sense of what we do support. So we support therapeutics, prevention, and diagnostics, because you need to know what you have in order to know how you're going to treat it. Since inception, since 2016, we have funded 77 unique projects in this space. By March, I believe that number will be 93. Today, there are 51 active projects. These come from 10 different countries all across the globe. And we've had 7 programs already graduate and move into advanced development towards patients. They do very responsive to those 2 threat lists that I mentioned. So critical and high, almost everything, urgent and serious. We target all the major syndromes. So respiratory tract infections, sepsis, urinary tract, sexually transmitted diseases like gonorrhea, skin and soft tissue and wounds, prosthetic joint infections, community-acquired respiratory infections, abdominal infections, and C. diff infections. And our diagnostics portfolio matches with that. We represent a lot of different modalities. So we do have your cell wall inhibitors. We have novel penicillin type molecules, novel penicillin binding protein inhibitors. We have a lot of things that also work on the cell wall, and also all those other mechanisms that I mentioned long ago. We also have a lot of really neat, crazy stuff that's never been proven in the clinic before. And I won't spend time here except to say, again, a lot of different things that we're trying here to really confuse the bacteria and be effective for patients with serious infections. And the same in prevention. We have vaccines, we have phage. You probably read about phage recently and its use in emergency cases. And then we have a small molecule C. diff program. And also, these are called live biotherapeutics, microbiome acting. You've certainly heard a lot about that. It's sort of yogurt on steroids, but very effective in terms of promoting good bacteria in the gut, and therefore suppressing bad bacteria so they don't breach the gut and cause sepsis in patients. And so that's all really great news. I want to end on even more good news, which is in July of this year, there's something called the AMR Action Fund that was created by all those big companies that left this space. They've pledged $1 billion, and I think they're now to almost $2 billion to take products where we end, which is early development, ultimately through to approval and commercialization. Why do they do this? Well, they do this because they want to preserve their other franchises. Nobody would go and have a knee replacement or hip replacement if they thought that an antibiotic wouldn't cure them if they had an infection. Think about all those cancer patients. They don't have good immune systems. You can't really treat them with your new innovative chemotherapies if you don't have good antibiotics. So this is preservation for their pipelines, but we're grateful for it. Governments are getting into it. Both the Wellcome Trust as a foundation and the US government are doubling down on their commitment to antibiotic-resistant infections as key foci in the next 5 years. And finally, both in the UK and in the US and hopefully around the world soon, the legislators are getting involved and trying to find creative ways to help support this market system. So in this case, the Disarm Act is something, you might not know this, but when you go into a hospital for anything, you're put in a treatment group. And that includes everything from the bed to the antibiotic, to the saline, to the nurse, to everything. And so if that package costs X, then the antibiotic can only cost some fraction of X. And so that really limits your ability to price these fairly. And this is legislation to maybe change that dynamic. And the Pasteur Act is very similar to UK Act, which is focused on like a Netflix subscription model, which says to companies, look, you're never going to market this thing. We're going to buy almost like a stockpile, and again, in a Netflix sort of way, so that we get the drugs to patients. We appropriately value you for the work that you've done, but we don't create disincentives in terms of high prices. We look at volume and getting drugs to where they need to be. And so with that, I'm done. I realize I took almost the whole time, but I'm happy to answer questions or have comments. Wasn't a lot about chemistry here, but this is how chemistry is embedded in something a little bigger. And I hope this was interesting for you.
Well, you may be up against it, not getting out of it that easily, because we have a lot of questions and we have stuff to give away. So let me go back to our first question from Bonnie.
Yeah, no, it's a great question. And the answer to that is no. These are two very different things. And so it's funny. People used to call this, and even our name, combating antimicrobial resistance. We and others did a survey of people saying, what is antimicrobial resistance? And people said, oh, I'm resistant to antibiotics. I have allergies. And these are very different things. So it's just your body reacting to the chemicals. This is more of a toxicity than it is a resistance development issue.
Here's a related question, I suppose.
That's a really good question. I don't know the answer to that question. Again, this is typically a so-called off-target pharmacology. So this has nothing to do with bacteria. This has something to do with another target that's a human target, that causes this reaction.
Okay. Here's the next. From your cousin.
Yes, absolutely. Hopefully, yes. So that is a big area of research, Michelle, both before you get into the clinic and then when you're in the clinic, which is asking, there's a whole series of enzymes that metabolize other drugs. And so what you don't want to do is be paired with something maybe that needs to be metabolized. And then if you inhibit that enzyme that does that work, all of a sudden you're going to have increased drug levels of the other thing and cause a toxicity. You can also have antagonism of antibiotics, but this should all be worked out well before the drug hits the market, and in fact be placed in the product insert, so that you know what is beneficial and synergistic, and you know what is antagonistic.
Here's a question from a mysterious person named the Walls Foundation.
Well, so I think hopefully, as that slide I took forever to get through shows, antibiotic resistance has always been here and will always be here. And so we recognize that your traditional direct-acting antibiotics have a role, and this is where again finding novel classes and novel ways to target these many mechanisms is important. But that's why I mentioned very briefly that we're also looking at a lot of non-traditional approaches. So a category, we had many categories in that pie chart, but one called antivirulence. And so what this is doing is actually focused on the toxins that the bacteria produce, not the bacteria themselves. We have other, approaches where we're harnessing the immune system. So we have a known antibiotic and it's hooked up to something that recruits the immune system. And so then you're getting sort of two ways. You're having the direct effect on the bacteria, and then you're also having the host effect. So a lot of things like that, as well as prevention. So we do feel that you need to have multiple ways, to target this problem in the future. I think it was two things. A lot of it, number one, there certainly was a thought, not widely shared. There's a lore that actually the Surgeon General in I think the 1960s said, the war on antibiotics is over. We can go do something else. But that's been debunked. He actually never said that. But in fact, I think people thought, well, we have a lot of drugs here. Let's go to work on some other problems. But then also there started to become this recognition that there wasn't the cost benefit for companies wasn't there. So you were spending a lot more money to develop than you could possibly ever recoup.
Here's a question from Dr. Ed, who I used to work with.
So the AIMS test is the test used to screen for, it's one of 3 tests to screen for genotoxicity, so which is what you don't want. So it's done actually in Salmonella, which is a bacterium, but it's looking at genotoxic effects rather than antibiotic effects.
Here's one from the mysterious Mary Dorr.
So there actually are a lot of non-private company groups that are engaged in this work. We actually are funding a group at the University of Queensland, so all the way around the world there in Australia, but they have a really good group there who have many different programs targeting bacteria, which is really great.
So just hold it for a minute.
Often the really cutting edge primary ideas for new targets or new classes or even new modalities are started at your major universities funded by the National Institutes of Health. And then that fundamental work often is translated into companies like the one where I used to work, where you really do the translation of that. So the new stuff has been established, and now it's your job to figure out how to make it work and do the multidisciplinary hard work of making a safe and effective drug.
Another question from Bonnie.
Yeah, definitely. So it's two things. So, yes, it is true that exposure to an antibiotic, the more the bacteria see it, the more, they have chances to figure out how to, I hate to sound like bacteria are a lot, well, they are alive, but they're that smart, but they are that smart. But so it gives them that opportunity. The other thing that happens, which is equally important, is when people get an antibiotic, Marianne Duffy, and they don't take the whole regimen. And so, you get a course for 10 days of therapy. 3 days later, you're feeling better. And then you put that in your closet for if you ever have you're starting to feel a little sick again, maybe you'll pop one of those antibiotics. Terrible idea. Take the course. Because in fact, if you don't run the course to completion, you're going to have this reservoir of bacteria that you don't eradicate, and they can in turn become resistant.
Okay. Here's one from Bill. Let's start with that, but there's another question.
Yeah, it's tricky, isn't it? It's very tricky. And so, yeah, I think there's a lot of emphasis now on One Health, which includes how and when antibiotics might and should be used in livestock for that very reason. So just the broad dissemination of antibiotics to improve the quality and shelf life of our meat products and plant products as well has certainly been abused and has been a component leading to drug resistance, no doubt about it.
Sorry, I'm writing down names for the big drawing here in a minute. But here's another question I wanted to get to. I think it's an interesting question. It's one that occurred to me too.
Well, this is interesting. So one thing I will note, I'm sure you've seen this too, is that the rates of regular flu, which is viral, not bacterial, but are really low this year.
Right, masks, right?
Like really low, and I think that has to do, again, a lot of this stuff is transmission, and if we're not going anywhere, we're not giving it to anybody. I would say the difference here with bacterial infections is, for instance, if you, cut yourself or you get a urinary tract infection, you will need antibiotics. And if you don't take them, there is a risk that you will end up with, generating resistance or, acquiring resistant bugs. So some of that's about transmission, but certainly, there are a lot of syndromes for which, again, wounds, etc., you can get all by yourself without interacting with anybody.
Yeah. Well, as you can see, my attorney is here now, Mr. Rooney. I brought him on board to go ahead and put those in to supervise the legality of. My assistant is helping her here. Okay. Of the drawing, no, that's not actually one. The drawing for the, well, let me hold it up for you. The beer chemistry pint glass that's so highly coveted that I think people are just dying to know who was going to win this. Now, Mr. Rooney please draw our winner this evening. Here we go. It's exciting, isn't it? This is my favorite part of the program. No offense to Erin and any other presenter. But thank you, Erin, by the way. Excellent program. Bill Ruther is the winner.
Excellent.
Tonight. So, Bill, you've won the chemistry beer pint, which I think you'll enjoy. We shall have a toast. And Mr. Rooney approves. Thank you, Erin, for an excellent program, and thank you everyone who attended tonight. I thought it was quite informative. A difficult topic to make accessible, but I think you did a great job. So thank you.
Well, thank you. This was really a lot of fun. I'm really appreciative of being invited.
Mary Ellen agrees. So does Erica, and your mother, of course. Thanks, Mom. So next week, Stacy Gonzalez Vargas will take us on a tour of physics from Newton to Einstein. From Newton to Einstein. That's it. Yeah. Okay.
Yes. And I just want to say, if Liam is out there listening tonight, I hope you received your books. And if you didn't, they should be coming any day.
Do you hear that, Liam? Your books are on the way. Okay, everyone. Thank you. And we'll see you next week. See you Tuesday and see you next week. Bye now.
Bye.